Sí. Good afternoon, everybody. We will start the next session. It is a pleasure for me to be here celebrating the anniversary of Giorgio. I have been working with him the last 25 years, and it is has been also a pleasure. Thank you, Giorgio. Okay, the first speaker is Felix Ritort from Barcelona University. We will speak about equilibrium breakdowns in single molecules and cell. Okay. Please.
Thank you very much, Juan Jesus. Good afternoon, everybody. It's a real pleasure to be here to celebrate the 17th birthday of a great person such as Giorgio Parisi. And everyone has to start telling something about his experience with him, okay? So I am a privileged one because my story starts in 1989, but actually when I started working with him was in 1990, when I came to Tor Vergata. He was at that time at Tor Vergata, and I was privileged because I was sharing the office with him, okay? So everyone, when you are very young, you are 23, you are put in an office with the other students, but he told me, non ci sono posti, mi dispiace. So he put me in his office, and I had a great opportunity to have conversations with him, and also I think I helped him a lot because many people wanted to speak to him. So what did they do? At the time we had BACS, which was a computer system. So they sent me a message, Felix, is Giorgio there? C'è Giorgio in stanza. He's in the room. I said, yeah, you may come now. And then they came and they looked for him. So all the time people was contacting me to know whether he was in the room or not, okay? And as you know, this is good because Italy is a country of favors. So if you give a favor to someone, then they can give it back to you, okay? But, yeah, mine for sure, yeah. So, going to the more scientific part, these are my first two papers with Giorgio, which I put here because I looked them this morning. I said, I like this work. This was beautiful work. Of course, these were my first papers. It was a lot of work, okay? So my first paper was doing something that we have heard about Victor Martin Mayor yesterday, and also Juan Jesus has been working a lot, which is numerical simulations of spin glasses. And my idol that time was Peter Young. Peter Young was so good making simulations in field, in finite dimensions, and we studied this hypercube, which is a many-dimensional single-cell unit that, of course, as an hypercube, as a single cell, has a number of sides of spins, which is 2 to the power d, and when you increase d, you increase the number of spins and you increase the dimension at the same time, okay? It's a mean-field model. And, but you cannot solve it exactly. Even if it looks at a very simple model, it's not like the Sherrington-Kirkpatrick model, which you can solve analytically. In the hypercube, you don't have links everywhere, and you cannot solve it exactly. But it's fully connected somehow. And we find this P of Q for the other parameter. I had another experience at the same time, which was the second paper. I work on a model which is called little spin glass model, which is like the Sherrington-Kirkpatrick model, but putting interactions between two, let's say, 2 bipartite lattices. So it's the same Hamiltonian as the SK model, J sigma sigma, but now you have sigma and tau, and you make connections J sigma tau, okay? Now, it was a hard work because the goal was to demonstrate that this is exactly the same as the Sherrington-Kirkpatrick model, but the demonstration is not clear at all. And what I was able to demonstrate is that at first order of Relpika symmetry breaking, the little model and the SK model give the same free energy. But for doing that, I had to diagonalize a very complicated stability matrix, okay? But at the end, I succeeded. And I was also, of course, I admired the talent of Cyrano de Dominicis and Imre Condor, who did this, of course, in the whole spectrum. I did just at first order of replica symmetry breaking. But that was very nice, this work in simulations and this work on theory. So what I want to say is, tanti auguri Giorgio, thank you very much for everything you have given me, not only to me, but all of us, okay? But now life is never perfect, and a few years later, I got corrupted by experiments, okay? And then I decided to start a career or to learn about doing experiments. Okay? So I had already an experience of 10 years in theory, first in spin glasses and in glasses. I said, no, now I want to see the truth. I want to see a nature. I want to see what nature shows me in an experiment. Of course, Giorgio is extremely smart, but I wanted to go further. And nature is an absolute master, okay? So what I'm going to show you today, and I go to the key, to the center of my topic, is about studying, if you want, disordered systems, statistical mechanics of complex systems, non-equilibrium physics, using the playground of small systems, okay? And what I want to demonstrate or convey to you is that small systems, when I think of small systems, I mean molecules. It can be molecules of 10, 20, 30, 40, 50 monomers, I don't know, but small systems at the molecular level also share all these properties that we are studying since the Sherrington-Kirkpatrick model and beyond. So they are a fabulous playground to investigate the problem about disorder and fluctuations, okay? So one of the problems I have been working over the past years, and actually was introduced by, in the previous lecture by Stefano Ruffo, he talked about these non-equilibrium steady states, so systems that to keep them on, you need to supply continuously power, okay? And these systems are very interesting because they are time stationary, so the properties do not change in time, but still they are out of equilibrium because you pump some energy, which in average is taken out to the bath in the form of heat. So they are continuously producing entropy. They are out of equilibrium. Of course, they cannot be described by Boltzmann-Gibbs distribution. So there are different examples. One is a current through a resistance. There is a Joule effect. You need a source of potential. And then you have the steady state of the electrons flowing through the resistance. You can also produce a non-equilibrium steady state by taking a fluid, which is in equilibrium, and then shearing one plate with respect to the other at a constant speed. You exert an external power, which is the average force due to friction times the velocity, and this is in average dissipated in the form of heat to the environment, okay? So these are isothermal systems. And the other ones are the ones that interest me possibly most, are related more to biological systems, which are, for example, chemical reactions that drive molecular motors across a substrate. And here is an example of a molecule called helicase. It's a protein that unzips DNA, separates it to strands, by using ATP hydrolysis. It's a steady state, okay? You open the DNA double helix continuously with a constant speed as a function of time. Another example is a living cell, and here I show you an example of a living cell, which is a red blood cell, which is not living because red blood cells are not considered to be alive. Okay? They are just, if you want, chemical bugs that experience some metabolic processes and they evolve, but they don't reproduce, they don't duplicate, and so on. So they are not considered living. But these are more close to active systems, possibly living systems. But still, these use sugar from the water in the solution, the glucose, and there are metabolic processes inside the red blood cell that make them maintain some sort of metabolic state. These are very interesting also non-equilibrium systems, steady state systems for the reasons that I will indicate after. So one of the results that have been much studied in our field of disordered systems and spin glasses and so on is the fluctuation dissipation theorem, which I will remind just for those of you who maybe don't remember the details. It tells you that basically a system in equilibrium, fluctuations decay in a way indistinguishable, spontaneous fluctuations decay in a way which cannot be distinguished of an induced relaxation, for example, by perturbing the system. So if I take a particle and I move it, I push on it, that motion induced by the force that is pushing is indistinguishable from a spontaneous diffusive motion without force. Basically, it says that. And it connects the correlation function to the response function. This is a so-called differential form. And if you integrate the response and the correlation, then you get, of course, for the response the susceptibility, and it tells you that the integrated response equals the correlation. I have written in this way, okay? There are different ways to write it, depending how you define the correlation function at 0 and so on, but this is basically the thing. You have also the same relation in frequency space, and you have to relate the corresponding quantity in frequency space to the correlation function, which is a power spectrum, S of omega, to the equivalent quantity in Fourier space of the response transform, which is the out-of-phase susceptibility. So if you drive your system with a finite frequency, the system has a lag behind the driving force. This lag, okay, which is this dephasing, defines the out-of-phase susceptibility. And you can measure experimentally these quantities, for example, the power spectrum. Nowadays, there are a lot of experiments in beads in optical traps, and here even in Rome, you have experiments. For example, there is Roberto de Leonardo who does these beautiful things, okay? And he measures this power spectrum, and this is a typical Lorentzian. And now you can ask, okay, can I verify my fluctuation dissipation, the fluctuation dissipation theorem by using this very simple model of a bead in an optical trap? So this bead is an harmonic potential. It fluctuates. Of course, it's confined to this harmonic well. And I can do the experiment. Sure, I can do the experiment. I can take my bead, which is exerting this trajectory, and then I can just keep track of the position along one axis. So a bead in an optical trap is three-dimensional. I just track one coordinate, and then I measure the correlation function, which I define this way. This is just convenient. Usually you see xt, x0. I define this way because I want to match the correlation function to the susceptibility. So at 0 time, I want the correlation function to be 0. But it's just a matter of introducing this x0 square here. So it's basically the same correlation function you are seeing all the time, okay? So you measure the correlation function. It goes from 0 to a maximum value. And you can measure also the susceptibility. So the susceptibility is an integrated response function. So for the response you apply an impulse. For the susceptibility you apply a step. And then you see after the step how the position of the bead moves. So for example, I take my optical trap. I move by 100 nanometers the optical trap, and then the bead follows the center of the optical trap, dragged through water, and that trajectory gives me the susceptibility Q of t. And I put them, if the fluctuation dissipation theorem works, I should get both them equal, and this is what you see here. These are experimental data on the susceptibility of one of these beads, which is about 3 micron in water, and the correlation function divided by kBt. This is a pure exponential function because of the simplicity of the model. Because an harmonic trap is a linear problem. But would happen in any other physical system, the identity between two quantities, okay? So what we do then is do something that are called FD plots, fluctuation dissipation plots. That was a nice idea, actually introduced from what I know in the field by Leticia and Jorge. They introduced the FD plots, right? And you plot the susceptibility as a function of the correlation, and you get it a straight line with a slope equal to 1 if the correlation is normalized as a function of kBt. And these are piconewtons per nanometer, because the correlation is nanometers squared, kBt is piconewtons per nanometer. These are the units. So these are real experimental data, okay? This is for a bead in an optical trap, but I insist this is independent of the model system. It can be as complicated as you want, and I can choose, for example, what I call a two-state folder, a two-state system. So molecule, for example, that can hop between two states. So these are experiments we do in my lab with an optical trap. We have a molecule that can be spontaneously unfolded. You tension the molecule is always folded. But then what you do is you pull on it up to 15 piconewtons, and then the hydrogen bonds of the hairpin cannot withstand against the structure. Spontaneously starts to hop. It's an experimental realization of dichotomic noise. And you have these sort of signals that you can do in the passive mode. If you keep the position of the trap constant, but the force fluctuates, as you see here, or you can do in the constant force mode if you actively move the optical trap to keep the force constant, but then, of course, in this sort of feedback, the trap position fluctuates. These are two ensembles of the same problem. Okay? It's like doing an experiment with a relaxation-ionizing model. You can control the magnetic field, or you can control the magnetization. Controlling the magnetization is very, very complicated experimentally. No one can do it. This is the reason why everyone talks about changing the magnetic field. But in principle, why not? You could control the magnetization and see how the magnetic field evolves. This, which is very difficult in experiments for magnetic systems, for single molecules for my systems is possible. So I have another experimental window to access what I call ensemble inequivalence. And then you can measure again in this two-state system, you can measure the correlation and the susceptibility doing in the same way as before. I measure the correlation function, just the correlation function of this dichotomic signal, or I can measure the susceptibility by switching the optical trap, shifting the equilibrium between the two states, and seeing how the trajectory that is hopping from one condition, okay, changes to the other condition where the probabilities of the two states have been shifted. Because the free energy shifts a little bit when you move the position of the optical trap. It's like a two-well potential. You can shift it by moving the relative deep, the relative height of the two states. And again, you see that this is more difficult to measure for obvious reasons, because you have to keep track very well of the drift in the experiment. These are molecules that have 20 nanometers of length that open and close, and you are in a regime more susceptible of drift, instrumental drift, than from the bead. You can move from microns, basically, hundreds of nanometers. So you are much less affected by drift in the other case. That's another case where the fluctuation dissipation theorem works. And I want to tell you what happens if you put your system out of equilibrium. And of course, you know there are several types of non-equilibrium states. The aging state is one of the most studied, possibly by people here, because it's a natural relaxing state of a spin glass of a disordered system. But there are non-equilibrium steady states, which means what I showed you before in the case of the resistance, in the case of the plates, in the case of the chemical reaction, and so on. So the question here we ask is, can we experimentally test the validity of the fluctuation dissipation theorem? Okay. And the answer is yes. And someone will say, okay, this is trivial. We know that the fluctuation dissipation theorem doesn't work in an equilibrium steady state. Why you want to do this experiment? Because it's an experiment on a molecule. So I want to use my one molecule system to do experiments of what is supposed to hold for big systems in the thermodynamic limit. I have a very small system. And to me, it was not clear that we had, being so small, we had the resolution to see these things. Okay? Of course, in equilibrium, I show you that I can do it. So what I wanted to do it was in the case of non-equilibrium steady state. For equilibrium, I did for a molecule, but still the question remains of what type of, let's say, fluctuation dissipation theorem, or if you want quasi-fluctuation dissipation theorem, because it's not satisfied, what type of relation do you find experimentally? And this is work done in collaboration with Udo Seifert, and he had a student who came to Barcelona and doing experiments and work, and we somehow collaborated doing theory and experiments together, okay? So the easiest way I wanted to study system, the easiest way to implement a non-equilibrium steady state has been introduced by several people. Giorgio Ciliberto actually did some of the experiments in the beginning. If I take an equilibrium system with a control parameter, the magnetic field, and I start to oscillate the magnetic field not as a sine wave, but stochastically with a finite time, so it's actually the control parameter changes dichotomically as if it followed a dichotomic Ornstein-Uhlenbeck process, then of course I don't have a natural frequency there. I have a natural timescale for the correlation, but I don't have a natural, let's say, an oscillation. I don't have a frequency. I have a timescale, I don't have a frequency. But still I am putting a timescale in my system and I'm perturbing it. I'm perturbing it because I am pumping energy with this oscillation, with this fluctuation. So I change dichotomically the signal between two values, and then I put my system in a non-equilibrium steady state. There are two important values in this experiment. You will see there is a force that changes dichotomically with respect to a baseline, a center line, Fc. So this is, let's say, the central force, and it fluctuates with an amplitude delta F up and down, okay? Plus delta F, minus delta F, and has an intrinsic correlation time tau e. So this force, which drives the system to the nest, has this exponential decay form defined with this excitation timescale. And the idea what we wanted to test was very simple, was the idea that we ask, okay, what happens if the excitation timescale is much smaller of this random forcing than the intrinsic timescale of the system? Then we thought, in principle, if this is true, the system should feel like if it is in equilibrium, but another higher temperature. Because relaxing on timescales that are much bigger, it cannot discriminate my external random forcing with intrinsic Brownian noise. Because intrinsic Brownian noise also has this timescale, but it is picoseconds. Okay? But conceptually, it should be more or less the same. But however, if I look at timescales that are bigger than the intrinsic relaxation timescale of the system, then I should have this form. And this form was introduced by many people. I list a few of them there that have contributed. But originally, this was done by two seminal papers in 1993, I think. at the same time nearly, okay? But the idea was that there should be like an effective temperature, which is also called fluctuation dissipation ratio, 4 times bigger than this time Ts. So somehow, after these times, you should feel this random forcing, and you should get a temperature that is different from the bath temperature. Then we did experiments on this system. Let me show you first. Well, in equilibrium, of course, you get this thing. And for the bead in the oscillating trap, let me show you how the experiment works. You have now this bead in the optical trap, and you have the dichotomic force, which is changing. This is the black line. So the black line is a dichotomic force. So how can I control the force in this experiment? Of course, this is a bead in an harmonic potential. I cannot control the force because the force is determined by the viscous friction. I cannot control that. What I can do is somehow mimic the force by moving the optical trap between 2 positions, which are about, in this case, 160 nanometers delta lambda. So plus 160 nanometers, plus 160 nanometers, minus 160 nanometers, and I can make the bead unhappy and crazy moving between the 2 harmonic potentials, okay, that are shifted. Now, what you see if you do this experiment is trajectory, okay, because when the position of the optical trap is here up, the bead wants to go to the center of the trap up. When the center of the optical trap goes down, the trajectory of the bead goes down. It wants to go here, but of course it cannot do it. It doesn't have enough time. And now this would give me, I could measure in this way the correlation function, because I have just the signal, the red signal would give me the correlation function for the position of the bead. At the same time, I can measure the response function in the non-equilibrium steady state by perturbing it. How I do it? So this oscillation, what I do is, at some point, instead of oscillating between A and B, I move A and B here and oscillate between A and B displaced. So there is a center of mass, let's say, of the position of the optical trap, sudden jump to a new position, and then the trajectory of the bead relaxes again to the new. So this region here gives me measurement of the susceptibility, because it is a jump, a step. It's not an impulse, okay? This stays forever. And so on, I come back to the original point, I repeat this many, many times, and collect a lot of statistics. So here is what you see, a trajectory, typical realization of the trajectory of the bead. Of course, you see that the bead, when it is in a position in a well, the bead oscillates between the center of the optical trap. And because this is a Hookean law, the force is proportional to the displacement. So if you fix the position of the trap, then the force follows this straight line, which corresponds to the stiffness of the optical trap. Now, at some point, what you do is you move the position of the optical trap rightwards or leftwards. Okay, it depends for you, for me. But you move leftwards, the bead goes to the right immediately. But pay attention, this measurement is not x, is not the position of the bead respect to the center of the trap. This is x, is a lap frame position. This is the position of the bead respect to the chamber. So even if I move the optical trap, the position with respect to the lap is the same. And therefore what happens is that this region doesn't change. But the force, because it goes to the other side of the well, changes, okay? And it goes from positive force to negative force. So this change in force, in Hookean force, means that respect to the well is, but you know, I'm moving the optical trap, but in the lap frame, the bead is in the same region, okay? And then you go up again by moving, by restoring the other state, and so on. Now, the important thing to understand is that if I do these sort of trajectories, I can measure the area, the circulation of these lines in the 2-dimensional plane, and therefore there is a net average area per unit time. So there is an entropy production. If you were to do the experiment without doing this stochastic driving, I would have also a very noisy signal, just if I don't move the optical trap across one of these lines, but there would not be net circulation. So the actual entropy production is the area, the average area per unit time or per cycle in these sort of plots. So whenever you make a representation of one force or one variable, like an observable, the magnetization or the position or whatever, and its canonical conjugate, okay, its corresponding force, if you make a representation of trajectories and you look there is a mean area per unit time, this means that you have a non-equilibrium state because you have a net entropy production. So here we measure the correlation function, we measure the susceptibility for different excitation times. So the average excitation time in this signal could go from a low value, 67 milliseconds, to 330 milliseconds. The intrinsic relaxation time of the bead in the optical trap, which is due to friction and due to the stiffness of the optical trap, is 200 milliseconds. So it's in between these two regimes. Now, it turns out that the susceptibility, you can make analytical calculation on this model and should not depend on any of these parameters. Okay? And here you see a slight difference, but this is experimental error, because we know analytically that it should not be like that. But the correlation function should depend a lot on tau e. So even if all these curves have the same intrinsic relaxation time, because it's the property of the optical trap, here is the property of the random switching between the two positions of the optical trap. There is a change in the correlation function. And now you can build the FD plots, and we were surprised that even for the bit in the optical trap that everyone has studied in the framework of non-equilibrium, let's say, experiments, the microsphere and optical trap is a very much experimental realization of a very simple Langevin model. This calculation had never been done by anyone. I don't have the time to show you the details, but basically we demonstrated that when this time tau e is less than the excitation time, the intrinsic relaxation time, when the excitation time was smaller than the intrinsic relaxation time, this idea of the excitation of the random forcing is true, and you developed an effective temperature. So you develop in K over C, you develop a line. So you initially start tangent to the equilibrium, so slope equal to 1 if you want, here in this plot, which is this equilibrium line, but you depart immediately, and only for times that are bigger than the time tau s, 200 milliseconds, you start to see these straight lines. Okay? So here the effective temperature would be for the red, which is 67 milliseconds, it's 5 times temperature. For the blue, is 7 times room temperature. Okay? So this is actually high because temperature is 300 Kelvin, so this is 1,500 Kelvin, and the other case is about 2,000 Kelvin. Okay? Now, if you go to a condition in which the excitation time is longer than the intrinsic relaxation time, there should not be, this idea should not hold. And actually what you find, and you can demonstrate analytically, is that there is no straight line behavior. Everything is curves always. Okay? So this is an experimental realization of a simple model. And then, to make the story on this part of my talk relatively short, we went to a more complex system, and we investigated a molecular two-state system, okay? Which is a molecule that holds between two states with force. I showed you before the experiment, and here I can do the same. I can switch the force dichotomically, and I can see in this condition whether there is entropy production, whether I am out of equilibrium, and I can plot the force as a function of the position of the optical trap. And again, the molecule, suppose that the molecule is at a given force, okay? And then if the molecule spontaneously unfolds, the force drops. If the molecule spontaneously folds, it goes up. But also I can move the position of the optical trap, okay? And by moving the position of the optical trap, I can pick transitions from here to there. So I have, let's say, 4 types of state at a given position of the optical trap, like this one. I have 2 levels of force, folded and unfolded. At the other position of the optical trap, more tensed, for example, I have 2 other states. I have 4 states, and these 4 states are represented by these 4 points, and I have finite circulation. So I have entropy production, and I can take my trajectories experimentally measured in equilibrium. Then I have this gray line is the position of the optical trap, which actually here what I'm controlling is the force. I do a force feedback, okay? Because it's very difficult to control the force if you don't impose a force feedback for a long time. So this is basically the force imposed on the molecule with 1 kilohertz frequency. So I keep a condition which is constant. The molecule hops between 2 states, and then I produce my nest by switching the force between 2 values. Let's say 16 piconewtons and 14 piconewtons. Delta F, I don't know, is 1 piconewton, and the center is 15. Switch back and forth with this stochastic switching, which has an average time, in this case, of 50 milliseconds. And the molecule also feels the change in the force, so the response in the hopping between the 2 states is also in there, in this red signal. And then I can do the experiment where I perturb the non-equilibrium steady state by shifting the 15 average piconewtons to another value, okay? And then I measure how these trajectories, when I do this change, this red trajectory changes. then we studied. We always wanted to compare with this intrinsic relaxation time respect to the excitation time. The excitation time here is 50 milliseconds, and we studied two molecules. One molecule which is slow, so compared to T, so 120 milliseconds, meaning that the switching is faster than the hopping of the molecule, and another case fast where the switching is comparable. Okay, in this case is comparable. Moving in these timescales is difficult because you have to find conditions where one hairpin has a given timescale beyond or above, and you have to design the structures accordingly. Anyway, the important thing is that when you do the measurements of the correlation and the response and the integrated response, you find this. We developed also a theory for the 4-state model that works pretty well. But then you see, for the system in which you had the slow hairpin, so the system in which the switching, the random switching force can be considered to be an aided Brownian motion, then we see a straight line with an effective temperature which is twice the room temperature. But in the case of the fast, we see the curvature and there is no effective temperature. Okay? And you can go even farther. You can go from the small to the mesoscopic, and you can imagine that you don't take just a hairpin of 20 base pairs, like in the previous case, it's short, so this is 20 nanometers in opening. You can take a molecule of 6,000 base pairs that opens like 7 microns. So it's really big molecule. So here I'm approaching the macroscopic limit, and you want to see how you go from the small molecular nanoscale to the much larger scale, micron scale. Because see, this 6 microns, this is the molecule, how it would open sequentially from the beginning of the stem to the end. This is 7,000 base pairs. And now what I do is again the same thing. I do a stochastic forcing, a random switching of the force between two values, and then I produce, okay, all this sort of noise and structure. Okay, so here is the tail of a trajectory. Well, this just wanted to show you that the molecule unzips and rezips reversibly. This is something very nice, because it shows you that the molecular level you can have reversible behavior, even if it is 7 microns long. So it's like if you take a piece of chalk, you break it, you connect it, and then you reform it again. Okay, so that's very interesting. And then, oh sorry, what happened here? Okay, I think I put too much weight in my presentation, too many slides. But what it shows you is that when you do the experiment, in that case, it seems also, because you are in the condition that the excitatory time and the intrinsic relaxation time are very well separated, and in this case, I think here is the figure, the excitation time was 2 milliseconds, and the intrinsic relaxation time is bigger. So let's see if this goes here. Okay, here you see the trajectory. So the intrinsic, so the excitatory time is 1.3. Okay, 1.3 seconds. Okay, so let me just. I don't know what's going on. It worked. I mean, I tried it. Okay, so it's 1.3 seconds excitatory time, and the intrinsic relaxation time is 2 seconds. So again, you are in this condition, and then you can generate the same thing, FD plot with effective temperature, which is 200 times. So when things got bigger, so this means that these effective temperatures can be actually very, very large. So let me just make a mention that this thing that we have done in non-equilibrium steady states, we would like to do for aging states. Now the question emerges whether you can do this sort of aging experiments with single molecules. So it turns out that single molecules do not want to age. Either they fold or they misfold, but you never see them aging to the non-equilibrium states, to the equilibrium state. It's very hard, and this is something which is very common, and it may have a biological meaning. This means that molecules have to be functional, so they have to either fold or misfold. You cannot have them slowly relaxing. But there may be exceptions, and one exception is something we are doing in my lab. I show you here. This is a work done by Aurelien Severino, which is a very talented student in my lab who is in Switzerland, and Paolo Risone, who is working also in the lab working on RNA, okay, and he's a student of Giorgio. He's here, he's in Barcelona since 1 year, and this molecule is an RNA that has a complicated structure because it has these non-complementary base pairs, it has bulges. It is what is called now an RNA thermometer. It has been discovered that many illnesses, like for example, meningitis, occur. So meningitis basically is an illness by which when you get fever, you don't deactivate, you activate the whole response of the virus. And this is because this virus, so it's supposed that when you increase the temperature of the body, it should kill the bacteria. But happens something different. This virus, or these bacteria, when they change temperature, they activate a response inside their bodies, inside their bacterial capside, by which RNA molecules change conformation upon changing temperature, and by changing conformation start to produce some proteins that are more resistant to the increase of temperature. So they modify, the RNA is like a switch, changes conformation, and then it starts to expressing some protein that at room temperature, 37 Celsius, was not produced. So you go above 40, they start to produce, and then even if your body increases temperature, the bacteria become more resistant because they start to have new molecules that can fight increase in temperature. They are more aggressive and they kill you. And meningitis is one of these. And this is this case of this molecule that has a transition to another conformation. And because it has another conformation, it has the possibility to have other states. And when we see is that when we study this molecule and we quench from an unfolded extended state to the native state, we see that the native state is reached, okay, but only after some time. And the fraction of native molecules as a function of time follows an exponential behavior with a time scale, this is seconds, time in seconds, of 13 seconds. So this molecule falls in 13 seconds, but 13 seconds for a molecule is huge, because molecules fall in microseconds. Say, oh, this is not the age of the universe like in a spin glass, but still 13 seconds allow you to study aging, non-relaxational properties in this time scale. So this is a curve, a typical trace. When you switch the force, the extension shows this low relaxation. This we are studying now. I will finish my talk because I think I have 5 minutes left already. So just mentioning that the same things, so you see molecules are very interesting, but cells are even more interesting. And one thing we wanted to answer is whether physical aging and biological aging are the same or not. So the fact that, of course, cells age, and they have mechanisms for that, and they die. Okay? Also red blood cells, which are non-living cells, they have an average lifetime of 1 month. So they go through the spleen many times until some time they change their structural properties, and some macrophages in the spleen see that they have changed it, and then they remove it, and then new red blood cells are secreted in the bone marrow, and then you start the cycle again. So red blood cells, even if they are not living cells, they have an age. Okay? And the question is whether we could investigate whether discriminate biological aging from physical aging. To make the story very short, you can do the same experiments when pulling molecules by deforming the red blood cell. And this has turned out to be a very beautiful experiment, because what you can do is exactly the same as for the molecules. You can measure the mechanical response of the cell across pulling, stretching, and releasing cycles. These are examples of different red blood cells, just curves. So there is strong heterogeneity. There are not 2 red blood cells identical, although there are 2 single molecules identical, because molecules have not the variability through sequence that a complex object that these may have. But we discovered something nice, which has been also reported by other people recently, that is, if you cyclically repeat the pulling experiment over the same molecule many, many times, okay, for the same force up to 20 piconewtons, the cell shortens, so it becomes more hard to deform. So there is a stiffening observed time scales of hours. And we don't know if this is biological aging or not. But certainly there is a time-dependent phenomenon. So on top of the non-equilibrium steady state, because this is hydrolyzing glucose all the time, or not hydrolyzed, but yeah, hydrolyzing glucose all the time in the glycolysis process, at the same time is a stiffening. And then we took these cells and we did a lot of non-equilibrium experiments. Okay? And just to make the story short, we start to play with this as if it were a magnetic system, putting a magnetic field, but using the force, taking it down, see how it relaxes. This is what you see, for example, if you do many force jumps up in a protocol. So you start from 10 piconewtons and you switch up to 20 piconewtons. And you see, and you keep then the force out. I mean, you just leave the red blood cell and the force decreases slowly. Then you go from 20 to 30, and you see a new relaxation of the force. From 30 to 40, so you do a step 10 by 10, and you have this very interesting relaxational behavior, which is very complex, by the way. This is fitted to 3 exponentials, okay? And to make the story short, we did many experiments, switching up, switching down, switching right, switching left, everything you can do, deforming, many type of deformation protocols. And we always saw these 3 time scales, so we could fit our curves with 3 exponentials plus a stationary value. The interesting thing is that all the experiments we did, okay, all the protocols demonstrated that the relaxation time, there are 3 intrinsic time scales in the red blood cell that are independent of how you deform it, okay, which is 1 second, let's say, 10 seconds, and 100 seconds. Okay? These are the time scales we could reach. They are very well defined. This is a very stable system. It's like you don't believe when you see it in the experiment. You say, how is that possible, red blood cell? I have not found any soft matter system, artificial, let's say, synthetic, that shows this behavior, okay? And then not only that, you discovered that even if you deform by 50% of the length, the linear response theory works. So the amplitudes are proportional perfectly to the change in force. Okay, you have linear responses everywhere. And this is not only my experiments, also in the experiments by Hayes Woute in Amsterdam with acoustic force spectroscopy. He deforms up to hundreds of piconewtons with acoustic forces and see this linear behavior. This astonishes me how much you can go. Of course, in these systems you could ask the same question, whether there is equilibrium breakdown. Of course, it is something we're investigating now. I just wanted to tell you this has been done, okay, for cells, for red blood cells actually, by Timo Betts, but at 0 force. I'm doing the experiments now at every force in a very wide range of conditions, okay? And we want to measure, not in frequency space, in real-time space, but it's basically the same. Now we're doing the experiments. This is a collaboration with Andrea Grisanti, which is somewhere there, okay? And Hayes Woute in Amsterdam. Okay, we are trying to apply our theory in this. And you can also do experiments on memory effects. I won't show you much about this, but these have to be properly designed because we cannot quench temperature. We can just change the force. And then this adds some sort of problems. It seems anyway that there are strong memory effects. So you do jumps in force from 20 piconewtons to 21, and then you measure the response in different times. You can superimpose them in a single curve. It remembers how much you deformed previously. These are the conclusions, but of course this talk was in memory and dedication, sorry. Sorry. Dedication to Giorgio. Thank you very much. I'm done.
Thank you very much for your interesting talk. We have time for a couple of questions.
Scusami, Giorgio. No, because to me memory is remembering only, but not because you are not there, of course.
Were you able to correlate the age of so can you somehow control the age of the red blood cell and see if there is a correlation with the fluctuation, dissipation parameter, etc.?
So that's a good question. That would be very nice, because you could prepare your sample at different ages, right? But the problem is that when you take a sample of blood, it's very difficult to, you take blood because because these are from healthy donors, so this blood is real blood. So they come at different stages. They have been produced in the spleen, the bone marrow at different ages. You cannot, it's very difficult to separate this, the age. And there are ways, but it's not trivial, and it's hard to see that. What you could do anyway, you could take a population which is more aged than the other one and start to probe it and see whether there is a change. So even if you cannot separate by ages, you could still see if there is a, let's say, an age, intrinsic age in a population. Okay.
So you mentioned very fast the ensemble inequivalence between constant force and constant length. So is it established, or which is the origin, or so is it the finiteness of the system that induces this property?
So it's known that constant force, let's say, and constant extension are not the same. Okay, it is well known. And in magnetic systems is the same. Constant magnetic field and constant magnetization is not the same. What I said is that at the experimental level, I can do in my case, whether in the magnetic case or in the electric case, is extremely difficult. So in my case, of course, I see strong differences. However, if these relations, fluctuation, dissipation, so on, are generic and hold whenever you have a non-equilibrium system, it should not depend specifically on which parameter you really control and which one you don't control. Okay? So which one is fluctuating or which one it is not, which means which ensemble you are. At the end of the day, it's the same. Although the specific mathematical form of the fluctuation dissipation theorem or the form of the entropy production or whatever it is, it depends on the ensemble. And that's what we have seen, that all ensembles are equivalent in the sense that the relations hold, but they give you different results, of course.
Okay, let's thank Felix again for this third thought.